friends, welcome back to another tutorial of Usmle topics. Today I'm going to be talking about glycolysis, and the reason I'm going to be talking about glycolysis on this one is because this is a series of videos which deals with cellular respiration. And obviously I'm only going to be doing cellular respiration of a human body, not of a prokaryote, only a eukaryote. And so when we talk about cellular respiration, we should be thinking about glycolysis, Krebs cycle, oxidative phosphorylation, or substrate level phosphorylation, either way you call it, and pyruvate metabolism. So this particular video is going to be dealing with only glycolysis because there's so many things I want to talk about. Secondly, I will try to make this video very dynamic in terms of information, so that I can kind of put things together for the different kinds of questions that can come on the real exam, so that this is not only about the pathway, it's about incorporating everything, or at least I'll try to incorporate everything that I know into this one pathway. So it's very, very important. So let's get started on the pathway. So before I talk about glycolysis, there's certain things that's very, very important to understand, and that is what is the primary source of energy in a brain, heart, muscle, and RBC. Okay, that's going to be my first question. What's the source of energy for brain under normal conditions, heart under normal conditions, muscles under normal conditions, and RBC under normal conditions? Okay, so it's kind of written here. So for brain, the primary source of energy under normal condition is glucose. Okay. For heart, it's not glucose, it's free fatty acid. For muscles, it's glucose. For RBC, it's glucose. Okay. So what about the next question? What's the source of energy for each one of these organ, system, and cell when the body is under stress? Okay. Again, I have it written down here. For brain, it's glucose. Again, for heart, it's glucose. It has switched from free fatty acids to glucose. For muscles, it's free fatty acids. So there has been a switch here. And for RBC, it remains glucose. So last but not the least, what is the source of energy for each one of these when we talk about extreme stress? Under extreme conditions, for brain, the source of energy is ketones. For heart, the source of energy is glucose. For muscles, the source of energy is still free fatty acids. And for RBC, it's going to be glucose. Now the question is, of all these, only RBC uses glucose throughout. Okay? What's the reason for that? The reason for that is RBC has no mitochondria. All right? It only can take part into mechanisms or pathways that take place inside the cytoplasm only, and glycolysis is only cytoplasm. It only takes place in the cytoplasm. So that's why RBC has no choice, because it has no mitochondria, it has no nucleus, it has to just survive on glucose no matter what it is, no matter what's going on. Okay, so that's why for RBC the whole thing is just glucose. So quick tip here, if I asked you what are the 3 pathways in the body which uses both cytoplasm and mitochondria, what would be your answer? I have a mnemonic for it, and I use the mnemonic HUG, H-U-G, HUG, and H for heme synthesis, U for urea cycle, and G for gluconeogenesis. Right? So these are the 3 pathways in the entire body which uses cytoplasm and mitochondria. All right, now let's talk about one more thing before we move on to the pathway, and that is kinase and phosphorylase. What's the difference between a kinase and phosphorylase? And the reason it's important is because all the time you're not going to know what pathway they're talking about. They can only talk about, okay, in an experimental experimental model, a kinase was used. So it's very important to get into the habit of learning these enzymes and what exactly they do, rather than just knowing where the enzymes are used. So what's the difference between a kinase and a phosphorylase? The difference is kinase uses ATP and phosphorylase uses free phosphate. Okay, so they both use phosphate, but kinase use in the form of ATP and phosphorylase use in the form of free phosphate. Okay, so now that we have that out of the way, let's start talking about glycolysis here. All right, so glycolysis obviously starts with glucose, right? And glucose uses an enzyme called glucokinase, okay, or hexokinase, either one. Uses glucokinase or hexokinase to make glucose 6-phosphate. Okay, glucose 6-phosphate. So what's the difference between glucokinase and hexokinase, and where each one of them are found? Glucokinase is found in the liver, hexokinase is found in everywhere else, okay? But glucokinase is primarily found in the liver. Now, one thing that we have to know here is glucokinase has high Km and low affinity. Okay? Glucokinase has high Km and low affinity, and hexokinase is the opposite. Okay, it has low Km and high affinity. What does that mean? It means that for glucokinase, you need lots of sugar, lots of glucose to stimulate glucokinase. Okay? For hexokinase, not so much. So obviously it makes sense, because so much sugar moves to the liver right after a meal that we don't need, if it gets stimulated with the low Km, then it's going to be always stimulated. So we don't want it to stimulate glucokinase. We don't want, because it has so much source of energy. But hexokinase, we want it. Everywhere else we want to pick up as much sugar as possible, with liver being our main source of glucose. Okay, so if I asked you now, how long does a plasma glucose last, and how long does liver glycogen, or source of glucose, last? Plasma glucose lasts about plasma glucose lasts about 2 to 4 hours, and liver glycogen lasts about 24 to 48 hours. Okay, so that's that. So once our glucokinase or hexokinase converts glucose to glucose 6-phosphate, it becomes this energy-rich compound, and it becomes trapped inside the cell. Okay, it becomes trapped inside the cell, and it cannot leave the cell. Now it has to kind of go down the pathway. So we talked about how we use ATP to make glucose 6-phosphate. Yes, it is a very, it's going backward, because glycolysis is meant to make energy. Yes, we are using ATP to kind of drive our direction down towards the end of the pathway. Okay, so from glucose 6-phosphate, the next step. So glucose 6-phosphate. Now the next substance is fructose 6-phosphate. Okay, so glucose 6-phosphate gives us fructose 6-phosphate. Okay. Now, I will not talk about every single step, so that is a step I'm just going to skip. If you want to know what's the enzyme and what's related, you have to look it up yourself, because there's a lot I need to be talking about. I want to skip to the next one, where fructose 6-phosphate is converted to fructose 1,6-bisphosphate. Certain steps of the pathway is not as high yield in the USMLE, so that's why I'm going to be skipping to this step. So fructose 6-phosphate gives us fructose 1,6-bisphosphate, and the enzyme that it uses is called PFK1. Okay, phosphofructokinase 1. Now, this is the rate-limiting enzyme. It's a rate-limiting enzyme. Since it's the rate-limiting enzyme, it's the slowest step in the pathway. Okay, it's the slowest step in the pathway. And if I asked you what kind of curve a rate-limiting enzyme makes, it's going to make a sigmoidal curve. So anytime they give you a rate-limiting enzyme and they ask what kind of curve they're going to make, you have to think about that it's a sigmoidal curve. So it slowly kind of picks up. It takes the most amount of time. So now, once we have fructose 1,6-bisphosphate, and let's say the pathway goes in that direction when we have lots of fructose 1,6-bisphosphate, something else happens. We have fructose 2,6-bisphosphate. So when we have fructose 1,6-bisphosphate building up in the body, we have also from fructose 1,6-bisphosphate, we are getting fructose 2,6-bisphosphate, and that is achieved by the enzyme PFK2. Okay, that is achieved by PFK2. Now this process, getting to fructose PFK2 and getting to fructose 2,6-bisphosphate, it's like when we have so much sugar coming in, the rate-limiting enzyme is slow and it can't keep up with making fructose 1,6-bisphosphate. That's why it kind of shuttles sugar in that direction, kind of holding the process a little bit, holding it off for a little bit longer. And this is another way. So when we have lots of fructose 2,6-bisphosphate being made, this is also a regulatory step, because fructose 2,6-bisphosphate in turn allosterically stimulates fructose 1,6-bisphosphate. So fructose 2,6-bisphosphate allosterically stimulates fructose PFK1. Okay, so here is another. Now I'm going to be talking about something else which is very, very high yield. Oftentimes in the step questions, they will ask you, what is a regulator? What is a regulator of a certain enzyme? Okay, these questions are it just drives me crazy whenever they ask these questions, because you have to memorize it, and I'm one of those people, I forget to memorize these things, but I need to sit down and do it, and everyone needs to sit down and do it. Even though these questions are so obvious, sometimes I miss them because I don't remember it like rock solid. So if they ask you, what are the regulators of PFK1? What are the regulators? Your answer should be fructose 1,6-bisphosphate. 1,6-bisphosphate. So PFK1 regulator. Okay, so what does that mean? It means that whatever stimulates PFK1 or what inhibits PFK1. So for that one, I have a little mnemonic here, and the mnemonic I use is CAF, C-A-A-F. Okay, don't ask me why I use this mnemonic, but I do, and it kind of works for me. Now for C, I have citrate. For A, I have ATP. The other A is AMP, and the last one, F, is going to be fructose 2,6-bisphosphate. Okay, so so this means that whenever there is a citrate, I have to continue this video on my next one because I cannot make longer videos than 15 minutes. So I'll continue this on part 2 of glycolysis. Okay.